Hi, Bita. How are you?
Hello, can you hear me?
Yes.
Oh,
Yeah,
oh,
I can
wait,
hear
my first time using this.
you. Perfect. Can you please say your name for me so that I pronounce it right? Just in
Um,
case.
wow, this Bitao.
Bita.
Yeah,
Okay,
yeah, just Bitao is good. Thank you.
Bita.
Yeah,
Okay,
yeah,
perfect. I'm
yeah.
posting the link to the presentation now so you can see. So above our profile pictures, there's a link.
Mm-hmm.
So if you click it, then it should be popping up. Go to link and then you should see the slides.
Oh.
Can you access them? Do I have the right access? Let me check again.
Oh, yes, I do.
Oh, good. Perfect.
Yeah.
Perfect. Yeah. We will be starting in around 7 minutes, so you still have a little bit.
Yeah, sure, thanks.
I just wanted to make sure everything works a few minutes ahead. Hey.
So how does this work? It was we just like a normal presentation or more like
Oh, okay.
chatting?
So it's kind of a mix. So I will
Mhm.
start by introducing you to the audience, like where you're working and your PhD. And then if you want to add any information, please do. And then usually we start by, I think Victoria will be here. She will usually ask like a couple of interview general questions, if that's okay with you. And then usually it's like time for your research, the presentation, and then like a discussion, with questions. And then the audience can also, in the meantime, either raise their hand or post questions in the chat room. And I would read them out to you. So I'll monitor, and Victoria will be monitoring that for you.
Sounds great. Yeah, thank you. Yeah.
Yeah, sure. Well, thank you for coming and making the
Oh,
comment and everything. It's
oh,
really nice
thank
of
you for your interest in our work.
you. Thank you.
Yeah,
Yeah, I'll hope you'll enjoy it. And
yeah, yeah.
yeah,
And, and now you are so you are organizing this every week or like is it like a weekly base because you are organizing this like a, what's that called popular science or
yeah. So it depends on the week. So this week we have, I think, 3 rooms throughout the week, and then by the end, like during the holidays, we won't have any rooms, and then it's about 2 or so rooms a week, like about 2 or so a week. It depends. I have been doing it a lot this year, but next year maybe I'll do it once or twice a week. I have to figure out the schedule. It was very a lot of work this year.
yeah,
So
I can imagine. Yeah.
we had sometimes like every day a room because
Uh,
way more speakers said yes than I thought.
yeah. Oh, but that's good, right? So
So, but it has been really interesting. I think I learned so much this year in like all kinds of science fields that are not mine at all. I think I've never learned so much before, so
oh.
that aspect was good.
And what are you focusing on? What's your research focus? Or is it?
So I've been doing more research for my own company and then consulting than academia. So it's a lot with mental health related
Okay.
issues, and then for consulting in general, it's like figuring out what the the next research step should be and writing like like proposal what should be done next and where. So yeah, but it's been interesting. Before I always did academia research. It was kind of a new thing that I started doing. It's also interesting. I always like switching, and then you realize what you like, what you don't like about the different ways.
Yeah, that's true. Yeah, and now you have the chance to now because you're doing all this interview and stuff, and then you broaden your interest as well, and then you
Yeah, it helped me. For example, I did some work for a stem cell company. Yeah, so I actually had before some guest speakers that did related research. Yeah, it was helpful also. Yeah, you're right. That I was, reading up for preparing the room. So I had to study to prepare for the interview, and so I learned a lot. Yeah. Yeah, welcome everyone. We'll start in a couple of minutes. Feel free to share the room. I'm doing that actually now. If you think friends or people that you know would find this interesting, and yeah, the presentation is pinned on top of the room, so it should be free for everyone to access. And hi, Victoria. How are you?
Hello, Katarina. Hello, Bitao. Is that how we pronounce your name?
Oh yeah, it's good. Yeah, hello, Victoria.
Hi, thank you so much for coming. This is really exciting.
I already said that you will ask like a few interview questions before we start, so.
Okay. And you're fine with that?
Oh yeah.
It sounds like, thank you. I'm ants are very special to me too. And I was an only child for until I was about 11. I guess many people are their whole life, so it's inconsequential that part. But I feel like I was on my own a lot, and I spent a lot of time observing ants because they were everywhere. They were accessible. It's like our own wild kingdom, and they were easy to observe and so fascinating.
That's great. Yeah. Because I mean that's great because I think last time our what's it called our research interest is come from observation from our daily life, right? So if you are interested in seeing the ant, and then you just curious about them as well.
Yeah, that's also very significant because in terms of education, and education is also so important too, that I think that because of the regimentation in school, how we're told what to look at and what to think and then judged on it, I feel that doesn't help strengthen our innate observation powers, or even patience for observation. So yeah, I have to agree with you, and glad to hear you. Just solidify that idea that I have, that observation is just critical for good science.
Yeah, so Eric already posted the question, so let's start before.
Let's get into it.
Yeah, welcome everyone to Science Society, and of course, a special welcome to you, Vita. And to introduce you shortly, Vita, he's a researcher at the University of Copenhagen in Denmark, and at the Center for Social Evolution, section for Ecology and Evolution in the Department of Biology. And he also did his PhD in 2019 there and the title of his dissertation was The Genetic Regulatory Network Mediating Caste Differentiation in Ants, a Transcriptomic View. And he research interest includes genetic mechanism in division of labor, evolutionary systems biology, and evolutionary developmental biology in social insects. And he was in the lab of Jacobus Boomsma and Buyoyi Tseng. I hope I said that right. And he worked there as a bioinformatics engineer in BGI. He is proficient and he has a lot of bioinformatics skills and statistics, and he intersects these different fields, including molecular biology, which I think is a really interesting approach. So welcome, Mitang. We are very pleased that you're here.
Oh, thank you. Yeah, and just a little update about my current position, because I was doing a postdoc in at the University of Copenhagen earlier this year until June. So I now have actually moved to Germany. So now I'm in the
where?
Germany.
Yeah.
In Freiburg. It's in the southern part of Germany. It's a most sunny city. Yeah, but it's still quite cold here now.
Oh, that's so beautiful. I grew up in Germany, so.
Oh, you great! Oh, great! Yeah.
Yeah, yeah, but
city?
it's a beautiful part of Germany.
Oh yeah, nice.
Yeah.
Well, then you probably have a better German than I do.
I don't know. If I would have learned it at the age you learned it, for sure my German would be worse. So let's say that. I learned it as a little kid, so it's different. It's more intuitive learning. So, but I'll hand the microphone over to Victoria for our interview. Thank you so much.
Thank you, Katarina, and thank you again, Vital, for coming and spending some time with us. Science Society is so grateful that you're here. And yeah, Freiburg is beautiful. I've been there as well and spent some time there, and it's lucky that's where you can spend your time. It's got this great food too, and good hiking.
Oh,
so
yeah, true, definitely.
Hope you can get out a bit. They make it so accessible. Okay, back to our topic. So ants, super excited about this. What I would like to know, in addition to the research that you'll share with us, is if you can recall what your initial fascination was with science itself. Maybe it was ants, for me it actually was, I can say. But maybe you can think back in your life when you felt connected to science and just really fascinated, and you noticed that there was something special around here.
Yeah, I mean, I think when I was a kid, I always interested in science. But I mean, in the beginning, I think I'm more interested in physics, because I think there's this rule, you can go from one to the other, and then you can just do this kind of induction stuff. But then later, I got more interested in biology, and then I feel like there's much more fascinating. And also I think my interest in science is also because I have this kind of a philosophy that I think by understanding more about science, or more about our surrounding, the natural world, we will have a better understanding of life itself. I mean, not just about nature, more about the meaning of life. That's I think that's my early motivation to learn science. Yeah.
Okay, that's interesting. So what have you learned about the meaning of life from your
I yeah, I think it's quite interesting, because at least because I mean, there's a lot of kind of philosophical question, right? So for example, whether we have a free will, and also what is individuality, and also why there's a cooperation and competition in human society. All these things, I mean, you can connected to science and have an explanation to it. And I think one thing that I learned most during my work in social insects, because in the beginning my motivation to learn more about social insects is not, actually not come from observation like you, because I know there are some people who are really the nature observers. They go to nature, they see ants, all fascinating. But for me, I think my early motivation to learn more about the social insect is more about my curiosity about human society. So I was, in the beginning, I thought, okay, maybe by learning more about the social insect, I can learn more about human society. But then now I think one big lesson I learned is that social insects are not the same as a human society. So yeah.
I was just, yeah, or maybe we don't know yet. Maybe we are, you know, on you mentioned being interested in biochemistry and that aspect, and maybe when we know more about our biochemistry and how that functions and influences our behavior and choices, maybe we'll feel like we're just an ant colony as well. Because I was looking at Deborah Gordon's work, and she studies ant colonies and the behavior. And she had mentioned that there were some. She was doing work with the she was examining how her question was, can they change tasks within a colony? And she found that they did change tasks regularly, and was observing that it was the hydrocarbon coating on their exoskeleton that they were communicating about which tasks to perform and what was needed. And so I'm thinking about us, about humans and pheromones, and who knows what we're responding to. But I can't say that. Yeah, I don't know if ants seem like they love
Yeah.
each other.
Yeah.
So yeah, um, so yeah, so it would be great to now step out of your way and hear more about your work and your findings. And so I'll pass the mic to you and just let you know, Katarina and I are here to moderate questions. Sometimes people will put questions in the room chat, and we can share those with you. And later we could bring people on stage, and they may ask you questions as well if you're interested in having a Q&A. So again, thank you for being here, and I look forward to hearing what else you share.
Oh, thank you, and thank you for the introduction. So so I think today the topic is about toward the mental biologies of superorganisms. So as I mentioned in the beginning, that one big lesson I learned is that the ant colony is not just a society; it's more like an organism. And then I think during this conversation or discussion, I think I can convince you more about this. So I think the one because as Victoria mentioned that you have this observation about ants, and if we go to nature, we often see this there's a huge division for labor within the ant colony, and then we can sometimes we can see that there's a lot of individuality of the colony. What I mean is that there's a part-whole relationship in the colony, because you can see that there's a morphological division of labor. You see the soldiers and the queen and the workers, and then even within the among the workers, there are different morphology among them. And then you can see that the morphology of these different castes are specialized for their task. And I can tell you that in ants colony, there's a lot of division of labor there. There's some individuals go to do this hygiene behavior, and some will go to do foraging, and some will stay inside the nest to do this kind of nursing behavior. So then you can understand why I say there's a individuality of the colony, because you can see that the ant colony, each workers and each individual with, let's say, the individual within the ant colony, just like a single each cell within our body. And of course, people have been comparing the ant colony to a society. But however, if we look at the life cycle of an ant colony, we can see that the ant colony is actually more like an animal compared to a society, because there's a Weismann barrier there. Because in the beginning, if you look at the life cycle of ant colony, in the beginning, there's a founding queen. She has just got finished her mating flight, and she got inseminated, and then she will go down to on the earth and dig a hole and go inside and begin the colony. And in the beginning, she will only produce the workers, which are just like the soma cell. And once the colony became bigger, the founding queen, she will produce the future queens and males, and the future queens and males they will fly out seasonally and then establish new colony. So this life cycle for an ant colony is actually similar to the life cycle of an animal, because you can imagine that in the beginning the eggs and sperm they meet each other, become an embryo, and then the embryo they will develop and differentiate it into different somatic cells, and the embryo in the end will pass on the genetic information through the gamete, the germ cell, to next generation. So you can see that the life cycle of the ant colony are actually similar to life cycle of an animal. And William Wheeler is one of the most famous ecology in the early 20th century. He actually realized this long ago, and I just quote his word. He said, between the ant colony and the cell colony, which constitute the body of a Metazoan animal, and many of the laws that control the cellular origins, development, growth, and reproduction and decay of the individual Metazoa, are seen to hold good also of the ant society, regarded as an individual of a higher level, of a higher order. So what he means is that the ant colony and an animal body is actually can be analogous to each other, and there must be some shared rules of between these two levels. So this idea has later been extended by the Julian Huxley in his Individuality in the Animal Kingdom, and later in the John Maynard Smith and Edward Smith in the Major Transitions in Evolution. And recently, Boomsma, my supervisor, he go back to the history and he reviewed the concept evolution of this major transition, and he proposed that there's multiple domains of social evolution that go from a prokaryote to eukaryote, and then to the multicellular organism, and then to superorganism. So that means that the transition between multicellular organism to the superorganism is actually similar to the transition between a unicellular organism to multicellular organism. So in our lab, we are focusing on the concept of a superorganism. That means we conceptualize the ant colony as a superorganism, and we try to ask the question about, okay, if ant colony is a superorganism, what we will predict from it based on the comparison to the multicellular organism. For example, we know that in the beginning I told you that there's this kind of germ-soma duality in the ant colony. There's a queen and worker, which is similar to the germ and soma, and then as Wheeler mentioned, that there must be some evolution and developmental similarity between the ant colony and an animal. So that's what we have been doing. And one of the two work that we have published in the past few years, one is that we show that by using the transcriptome, I mean the gene expression level of the different caste, the worker and the queen caste of different ant species, and then if we try to look at the similarity of the transcriptome across these species, we can see that there's actually a similarity of the pattern between the different castes in different ant species and the different organs in mammals, because we can know that homology is one of the key concepts in evolution. For example, the four limbs in different in the four limbs in birds and humans, they are homologous to each other. So So we know that there's a genetic record network underlying this homologous structure there. And similarly, in ant society, we have this kind of a queen and worker, these two different castes. So these two different castes are homologous across different ant species. So we have proved this one. And then a second work that we have done is that we show that the insemination of a virgin queen, that is once the queen have been inseminated, the transition between a virgin queen to a founding queen is actually similar to the transition between an egg to an embryo. Yeah. So this is what we have been doing, what we have done before. And today I'm going to tell you more about the similarity of the caste differentiation process and cell differentiation process in animal. Because as I mentioned before, the ant colony can be conceptualized as a superorganism. So for each individual in the ant colony, you can conceptualize as a single cell in an animal, and therefore then we can analogous to the caste differentiation process in ant colony as the cell differentiation process. And Waddington is one of the founder of epigenetic study. He proposed this epigenetic landscape to describe the cell differentiation process during embryogenesis process. So our question is, can we try to use the epigenetic landscape to describe the caste differentiation process in ants? So because if we try to describe the caste differentiation process in ants under the epigenetic landscape, we can ask multiple very fascinating questions. For example, whether the caste differentiation, for example, like in cell differentiation process, you can recognize different cell type in the later stage, but actually the cell differentiations already occur before the morphological difference occurred. So therefore, we can ask the same question, for example, like in caste differentiation process in ants. In the later stage, there's a huge difference between the worker and the future queen. They're morphological quite distinct from each other. However, the caste differentiation process has already occurred before the morphological difference between castes occurs. So our question is that can we try to predict the fate of each individual before the morphological difference emerge? So to address this question, we have this kind of idea that we use this kind of a single individual transcriptome, because nowadays the single cell sequencing is quite popular, which people just isolate each cell within the organs and try to sequence each cell to see what is the composition of the organs and how cell differentiation process occur during the organ development. So our idea is that if we can do this kind of single individual sequencing, so we try to see each individual ant as a single cell, then we try to follow the developmental trajectory of the caste differentiation process. So with this idea, so we first develop an algorithm. We call them the backward progressive algorithm, which is used to predict the caste fate of each individual before their morphological difference emerge. So this algorithm is based on the idea that there is always a direction of the developmental process, because there's always the arrow of time. That means that the times always go from past to present and then to future. So we have this idea that we can actually use this process to predict the caste differentiation and predict the caste fate of each individual. So with this algorithm, we successfully identify some markers for early caste identity. For example, there's two genes we identify. One is called 39AA7 and the other is MYD3, we found that these two genes are specifically expressed in the germline of the future queen, because you know that in the ants, in some of the ant species, the workers are complete sterile, and then only the queen, they have the germ cells. And we found that the gene marker that we identify are specifically expressed in the germ cells, and only in individual have so this individual must be the future queen. So now we have identified we can predict the cast fate of each individual in the early stage, just like we are able to predict the cell fate of each cell. So our next question is that is the caste differentiation process canalized? For example, as I mentioned in the beginning, there's epigenetic landscape. That means that in Waddington, he proposed that during embryogenesis process, each organ they are gradually canalized into this distinct morphology. For example, you can ask the question, okay, for example, if you look at your arm and your heart, you can see there's no similarity between your arm and your heart, because these two are different distinct organs. So you can see that during the organ genesis process, each organ are gradually develop into this kind of distinct shape, which is for the function of the whole individual. So our question is the caste differentiation process similar to the cell differentiation process in animal, that each individual are gradually develop into this kind of distinct phenotype? So with this idea in mind, we may use our single individual sequencing, and then we calculate the developmental potential score, which are used to quantify how well each individual are developed into this distinct phenotype. And we can clearly see that during the caste differentiation process, the worker and queen, and the worker and the future queen, they are gradually diverge in their transcriptome, and then they are for workers within the for individual within the caste, let's say all the workers, they are gradually converge into a point, into a distinct transcriptome space. So that means that during the caste differentiation process, each individual they are gradually develop into this kind of distinct, let's say, destination for their phenotype. And we further compare the within caste variation, and we further compare the within species variation and the between species variation. Let's say that the within species variation, we can use it to quantify how well each caste are being canalized. For example, if in the end all the workers are develop into the same phenotype, you expect there's a very little variation among them. I hope this makes sense. And then by calculating the within species variation, we can compare the variation in variation difference in different castes. For example, we can ask the question like, is the variation in the workers are higher than the queens, or the other way around? And we found that the variation in the queen, the future germline, are actually much lower compared to the workers. So that means that during the caste differentiation process, the queen caste, they are actually a much more canalized. They have a much more distinct phenotype compared to the worker. And similarly, we can compare the between species variation, because previously I'm talking about the within species variation, which we compare the individual within the same caste, within the same species. But we can also compare the worker between different species. And surprisingly, we found that similar to what we observe in the developmental process, in the the variation in the worker class between in different species are actually also higher than the variation between the queen in different species. So that means that even in the evolution, even in the during the evolution, the germline, the colony germline, the queen are also more developmentally conserved compared to the workers. So this is very interesting. So we then let's ask the question. So what is the function of genes that are highly canalized? That means because we can see this overall pattern, seeing that okay, each class are actually the class phenotype are actually gradually canalized into this distinct phenotype. And our next question is okay, what are the function of the genes underlying this, and what genes are regulating the canalization process in caste differentiation? And we found that the juvenile hormone, because when we look at the gene expression profile during the prepupal stage, which is the stage for the metamorphosis. So metamorphosis means that they transition from the larva and then to the pupa. And we can see that genes associated with juvenile hormone are actually highly differentially expressed. So we further do this kind of manipulation process. We fit the workers with juvenile hormone, and then we also fit the future queen with a precocing one, which is a juvenile hormone inhibitor. And we see that once we fit the worker with juvenile hormone, the worker, they are becoming much larger, and then they actually can see this kind of small wing bud in the workers. And if we fit the future queen with juvenile hormone inhibitor, the future queen, they're actually becoming smaller, and they develop into this kind of intercaste phenotype. That means that because previously I mentioned that if you look at the muscle and the, for example, if you look at the let's say the muscle cell and the neuron cell, okay, that these two cell type must be quite distinct. However, let's say that if you add some chemical there, and then you can make like a mosaic, intermediate phenotype between the muscle cell and the neuron cell. So, and actually we are so similar here. So once we use the juvenile hormone, we can have this kind of intercaste, that is an intermediate caste between the worker and future queen. So that means that the juvenile hormone actually are regulating the canalization process in ants. And then the next question is okay, so caste differentiation process is canalized. So what is the function of these genes? So we look at what genes are being canalized, and we found that in the queen, genes associated with the gonad and wing muscle are highly canalized, whereas in the workers, genes associated with the brain are highly canalized. So this actually makes sense, because if you think about the function of this different caste, the workers they are performing different maintenance of a colony. For example, a foraging, nurturing the brood within the colony. So that's which require a lot of which shows importance of the neuron functions. And whereas for the queen, or let's say the future queen, she will, as I mentioned in the beginning, she will fly out to do the mating, and then the gonad or the ovary of the queen are actually a determinant factor for the success of the future colony. So that's why I say it makes sense to see that the wing muscle and the ovary related genes are highly canalized in the queen. So that also means that the canalized gene expression level is an adaptation, but this adaptation is not only for the purpose of each individual, but for the purpose of the whole colony. Just you think about, if you think about the muscle cells and neuron cells, this different cell type are highly canalized, but the canalized phenotype is actually for the purpose of the whole individual. Let's say our whole body, for our whole body to maintenance and then to pass on our gene to next generation. So we observe similar things here. The canalized gene expression level in the ants, they're actually also highly canalized, but the canalization is for the adaptation of the whole colony. And we further we try to look at, because here I show you these genes that we have their function based on Drosophila, but we also want to know, okay, what if there's a we also try to look at the first genes, the top genes that are showing highly canalized expression pattern. We try to ask, okay, what is the function of this highly canalized gene? Because this gene, when we found it, there's no functional annotation there. And then we do this in situ hybridization, which we use like a marker, like a fluorescent way to look at where this gene is being expressed. And we found that this highly canalized gene is actually expressed in the follicle cells of the ovary. So that means that this hymelateral, I forgot to mention this gene is a hymelateral specific. That means that this gene you can only find it in the wasps, bees, and ants, but not outside. And we found that these hymelateral specific genes are highly canalized and is expressed in the follicle cells. So this is actually consistent with what we found previously. We saw that genes associated with ovary function are highly canalized. So we can see that this hymelateral specific genes are also associated with the ovary, with the reproduction. But what is more surprising is that once we do the RNA, let's say lockdown, that means that we try to use a molecular method to make this gene has a lower expression level. And once we try to lower the expression level of this gene in the queen, for one thing, we found that, okay, once we lower the expression of this gene, the queen become less fecund. That means that the ovary becomes smaller. But what is more interesting is that not only the ovary becoming smaller, the body size of the future queen and the wings are also becoming smaller. So that means that once we lower expression of these genes, which is only expressed in the ovary, we also influence their phenotype of the whole body. So that means that these genes are actually have a different multiple function and maybe constraining the evolution process of Caste phenotype. So for that, I would like to conclude, because in the beginning I would like to say that, okay, I would try to I hope I can let's say convince you more that the ant colony is more like a organism instead of a society, because we can see that because we already see that there's a germ-soma differentiation in the ant colony. That's the future queen and the worker. And this germ-soma differentiation process is actually occur in the early developmental stage. And compared to the colony soma, the colony germline is much more canalized and evolutionarily conserved. And finally, we can see that the canalized gene expression level in the ant colony are actually adaptive at the level of colony. And we also see that the canalized gene expression might influence the evolution of a Caste phenotype. Yeah, so I think that's all. Yeah, and I would like to thank my supervisors and all our funding, and our collaborators. Yeah, because it's require a lot of work, particularly the molecular validation. Yeah.
Well, thank you so much for presenting so well this all this work, and quite complicated work, in a way that that we can all understand. This was a wonderful presentation, and also so interesting the work you did, how you show how this bigger organism organizes itself. That's really interesting. Do you, or did you analyze also environmental factors, how they then can they change like the ratio, or you know, does this canalization get affected by environmental factors? Because you said it's quite conserved, so.
Yeah, that's true. No, we haven't because because we are just try to do that in a control way, right? Because once you have a environmental factor, then maybe you'll be less canalized. Yeah, that's something that we haven't done. And also, I mean, canalization is like a what's it called? How to say that? It's like a phenomenon. And of course, how canalization, how canalized phenotypes being regulated is another question. Yeah, as you mentioned, environmental factor may also play a role, but this environment then depends on how you define environment, whether it's like a chronic environment or like a abiotic environment.
Yeah, and also what I think would be really interesting, I read at some point, because it was a project, I'm repeating myself for people that went to the last rooms, that I wanted to do with my kids one day was to see if like a caterpillar memory gets passed on to the butterfly. So would be also interesting here, how do you think that from the previous colony, let's say, I don't know, traumatic events such like a parasitic big animal coming and eat up half the colony, like, do you think they get passed on throughout this process, like colony memories like this? Since, you know, if you see one colony as an organism, we see in mammals and so on that like traumatic events or some significant memories can get passed on to the next generation. What do you think would happen?
Oh, that's a really fascinating question. I mean, yeah, I mean, and actually, we have some people. Some one of my colleagues, she's actually doing this. Yeah, so yeah, because I mean, so for example, like how so this is more like whether epigenetic can inherit to next generation, right? So for example, in this colony, maybe there's some starving, and then the queen, there's some influence on the queen, and whether this influence will impact the future queen she produced, and then this future queen will also have a poor or better colony. So I think, I mean, I think this could happen, and but then it depends on because it's two way, right? One is that of course there's no argument that once you have a poor nutrition of the current queen, of course she will produce offspring that will have an impact, because for example maybe will impact the nutrition level of her offspring, and then her offspring may will produce a poor or better colony. I don't know, but we can definitely say that there must be some effect there. But then there's also a question about whether this process is let's say whether it has been met frequent enough during the evolutionary process, so that this inheritance will become adaptive or not. Yeah, I think this is a very interesting question, and must deserve more research. Yeah.
Yeah,
May
thank you.
I?
Go ahead,
Go.
Victoria.
Yeah, I don't want to cut you off from your next question, just to ask another one of this, if that's okay.
No, yeah, I just wanted to thanks and pass on to you. Thank you, Victoria.
Okay, yeah, thank you, Vital. So I see that what you're saying is that maybe any phenotypic response that we see to a stress, it could be, for example, as you're saying, there wasn't enough food, and so that was expressed, lack of nutrition, and we can see that in different ways. And I'm thinking maybe, Katarina, what you were wondering maybe is if it's communicated somehow to offspring. And I'm thinking, you were saying environmental stresses. So I'm thinking, if there's habitat damage over generations, if because they're communicating, that's what they, that's what we say they're social. If there's something that they're communicating to future offspring, but how would we differentiate a response from deliberate communication?
What do you mean by deliberate communication? You mean communication from the worker to the future queen, or?
Yes.
Yeah, that's a that's a difficult question. I mean, yeah, I don't know. I mean, yeah, yeah, because I haven't thought about how to decide an experiment to test this. So yeah.
So I think one thing related would be that the ants use, for example, different, like from the dirt, different things to collect and transport liquid food to the nest, right? Some ants, they build, for example, their underground tunnels in a
Yeah,
specific way. And maybe they would have to change their approach based on long-term environmental changes. So maybe that moment, I think Victoria would maybe think of that moment when they change their method, if that gets taught or if it's something that gets like in, expressed by epigenetic changes or both, maybe.
Exactly. You really made me think, Vital, when you brought that back to the response, the phenotypic response, that what is it that we can deduce from what we see, and how important it is to make sure that that we come to with our questions with an open mind, ready to receive the truth of what's before us.
Yeah, I don't know. Like, what type of, do you have like a regulated, I would imagine, humidity and type of dirt you use for the experiments? Maybe if you switch the dirt and more
yeah,
water, they would have to build their tunnels differently. But maybe they just have the spectrum of behaviors already in their DNA, or maybe it's something they
yeah.
need to figure out and then pass
Yeah,
on.
but then also I will also ask the question about how frequent for them to meet this situation, because I really appreciate the question, because the question is more like for example, if there's something happening in the ant colony, will the worker respond adaptively to trace the next generation? But then the question is that is it occur frequently for natural selection to select this response, or because in the end
Yeah,
this must be adaptive, right? You are what you are
yeah,
trying to propose.
yeah. I think with the with the current climate change,
Yeah, but but the
yeah,
climate change is
situations
not
are changing.
is recent. Yeah,
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 so we
yeah.
can predict, will ants make it in those regions or will they all just die? Maybe you could predict with the work in the lab if they are adaptive. Maybe in the lab we could train a bunch of ants and then send them out to teach the others. I don't know. Probably not, but would be interesting predictive
Yeah, because I mean,
analysis.
because this epigenetic inheritance can happen, but sometimes only in species of meet this environmental traits frequent enough, and then they actually have this adaptive response. But then for something that they have never met before, then it's quite difficult for it to be adaptive. That's why I'm thinking, yeah.
Yeah, I don't know, the ants that when there's a flood that they make together some sort of ant island, so um the whole colony and the ants can survive. You know those videos
Oh yeah,
when
yeah,
there's
yeah.
a flood. Yeah, I feel that makes me kind of optimistic for ants, but I don't know.
Yeah, could be. Yeah, could be.
Well, what they really need to communicate is to humans how we can peacefully coexist. Because really, there's a flood, it rains, they come, they might walk along, the perimeter of the house, and then they're gone when the rains are done. So, but I guess then I'm thinking from what you're saying about the difference between communicating within a generation with with those hydrocarbons that are on their exoskeleton, just things that they can sense by whatever it is, their touch or smell, between that communication and epigenetically in between generations. I'm thinking, yeah, I guess of sea level rise, or in the case of monarchs, and their food isn't as available. How are maybe all social insects are coping in similar ways, if they are at that.
Yeah, yeah, could be. I don't know. I mean, for example, we know that now with the climate change, because for example, in the termites, they have this kind of natural fly, and also in the ants, they also have. And this natural fly is actually associated with the rain season. For example, when they transition between the dry to rain season, and during the transition, they fly out because only in the raining season they will have a more probability to survive the new colony. And now we know that, for example, in the Africa and uh, let's say Ivory Coast. In Ivory Coast, because this climate change, so then the rain season is actually becoming the first rain is actually becoming a delay, or maybe sometimes earlier, and then because these are becoming unpredictable for the termites, so they sometimes they fly out of season. So that's what I mean that I think this communication and within colony and this transmission to the next generation will need to be something that historically occur frequent enough for them to develop this adaptive response, and for something that is occur recently like the flooding and stuff, it's actually maybe quite harmful for them, because they still need time to 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 get the selection to to find the solution.
Yeah, yeah, this is such an interesting discussion. And the other thing is to come back to your previous motivation to learn about humans, individuality and social interaction, and to have basically an animal model. When you talked, I remembered that there were a few philosophers that made like similar comparison that our social structure is a organism. I think the most famous one is Herbert Spencer from Britain, but then there was actually a previous one, a philosopher that already wrote this in 1920, Comte. And it's really interesting to look at this analogy. Would you say it's just a more complicated, layered, and longer evolution of an organism, our social structure? To understand it, I think it's always useful to go back into easier models and get understandings from that. So do you still think we can learn a lot from those models?
Yeah, I think that's actually quite a good question. And then, as I mentioned, that my supervisor Boomsma, he has recently do this review of the development of the concept of organism in social insects, and you can see that he is actually quoting the Weisman and Julian Huxley, that is more than 100 years ago. And then at that time they already see the huge difference between the human society and the ant colony, because an ant colony is actually a family, and a human society is composed of multiple families. So that means that the human society is at most an organization, but not an organism. And if we are really quoting the early philosopher, you can also see that some philosopher, because I can tell you a funny fact is that in the early days, people do not know that the queen of honeybees are female. They thought that the queen of honeybees are actually male, so they think that, okay, because the honeybee have this organization with the king and then a lot of the workers, so maybe we should support the monarch. So you see, this early understanding is actually not. Sometimes they are not correct because they don't have a good understanding of the biology themselves. Yeah. So what do you, does it make sense?
Yeah, definitely. That makes sense. And the interesting part I thought that was that we kind of have that they theorized was we have this very complex brain because we created this very complex society, and we constantly need to adapt to it because with more complex brain we create more complex society, and then our brain has to grow again. I think that was interesting to explain basically why we developed such a complex brain. But yeah, I agree. There are big differences, and it's oversimplified, but still an interesting play of thought, I guess, to play around with. Yeah, I wanted to encourage everyone in the audience to post comments, questions. So yeah, please do so. We always welcome people on stage and ask their questions. And we because this topic I think is so interesting on so many levels, so I'll just keep asking questions myself if nobody else comes up. What I also think was really interesting is the time point of the when basically the superorganism starts to canalize. Is there so is the time point always the same? Or does it vary from, did you look at different ant species maybe? Or is it always the same for all of the different ants?
Oh, it's actually quite different across different ant species, because for example, here we have two ant species. One is the pharaoh ant, and the other is the leaf cutting ant. And for the pharaoh ant, we can see the canalization actually occur very early, and then for the leaf cutting ant, it occur much later. So this is actually similar, because as I do the comparison of the ant colony to the animal, this actually is similar to different animal species. For example, like the Drosophila, where the germline are also already determined in the oocyte stage, whereas in the mouse, the germline actually occur in the later stage, but it's still in the early embryogenesis process. But you can see that just similar to the animals, which have a different time point for determining the germline. The canalization process in different ant species also occurred in a different time point. And then I think this will also be a very fascinating question, I think, about, okay, so why there's difference in different ant species, or let's ask another question, why there's difference in different animals for this kind of germline determination time point? Is it adaptive, or is it just some consequence of the evolution process? Let's say whether because it happened in the ancestral stage, that's why they continue doing this, or is it because it's adaptive for it to survive? So yeah, I mean, there's difference there, but why there's difference is something really worth to study.
That's really interesting. that. That's so different. Yeah, I'm curious to hear. And for these in-between ants, where you change the hormone exposure and they're kind of in between the queen and the worker, what's their behavior? Like, did you get to let them become adults and check how they behave in a colony? Is that different?
Yeah, yes, I think there's difference there, but I didn't follow the behavior observation, unfortunately. Yeah. Yeah, I think we're actually trying to look at their behavior, and from what we know is that the that's just some from what I remember. I remember that the worker, they become a less go they don't go out so often. That's what I remember. But I think it will be worth a detailed observation to see what are what they're actually doing in the colony once they become this kind of intercaste.
Yeah, it would be really interesting. Also, how the other ants would behave and reacted. Maybe they would just kick them out because they are different, or or they just adapt, or maybe they just adapt to wherever they are exposed to. Which, if they're exposed to the workers, they'll just behave like the workers, maybe. But maybe the workers don't
Yeah.
want them.
Oh no, I mean, yeah. Or every cell is useful, right? Maybe.
Well, yeah, because they're taller, like they're bigger, right? Maybe they are very useful. They will become the popular worker. Will be interesting. Well, there's a lot of really interesting behavior ahead, like studies ahead to analyze, I think. So will be interesting to see what the future brings. So you're still working in this field with ants in your new position?
Actually, I'm still working on ants, but my now new focus is actually on termites, because I think there's a lot much more unknown in the termites. So that's why and also, I mean, as I mentioned, this superorganism has convergently evolved in multiple lineage in termite, in bees, and in wasps and ants. So I think it'd be interesting to also look at the termites, because termites is actually much very far away from the ants. So I want to see whether what are the similarities there, and then what are the difference there.
Well, that will be really interesting to compare. So I'm really looking forward. Maybe when you have some results in a year or two, you could come back and talk with that. That would be like talk about that research that you're doing now. That would be really interesting. And yeah, we wish you all the best for the future, for your future, and for your research, and that you get a lot of grants. Uh, and I see that we have two people. If you still have maybe a couple of minutes, I know we've going on for an hour, to answer Carl's questions and share your question. Welcome to the stage. And if you have a question for our guest speaker related to his research, please go ahead and ask your question.
Uh, I'm Sherry. I'm from Guadalajara, Jalisco, Mexico. And my question is, how you make your medicines stronger? Cause I make poppy field heroin from Sinaloa.
We are talking about ant research, so if you have a question about that, please go ahead. If not, we'll skip to Kyle. Kyle, welcome. Go ahead.
Yeah, I was kind of multitasking there, but I wanted to take some time to just come up here and say thank you. The philosophical implications here of your work are quite immense. I heard Katerina bring that up. I'm thinking about like Michael Levine and his work with xenobots, and just kind of like organisms and biology and AI and the morphogenetic code that he coins it. So he'll look at systems like this, and instead of just like, this gets into a little bit of, where does the mind extend to kind of sort of thing with Markov blankets. But it seems like the ant community is almost working as a self, such as like other. Um, I guess I should actually look at what the term is: superorganisms, the next level of biological complexity above that of the organism itself. So I was wondering if anything about that came to your mind, or maybe altruism. I remember that I heard a great question that was asked by Joyce about humanity and working together. So the kind of idea of altruism came to my mind. So I was just wondering if anything came to your mind about like um, not looking at the community, I guess, or the ant colony as just one group of ants piled together, but also looking at the colony almost um, as one kind of entity, and then also just kind of about altruism.
Oh yeah, thank you for the question. I mean, I think yeah, that's exactly. I mean, because as I mentioned, the ant colony is like a superorganism. So that's what it means that we see the ant colony as an entity, and when we talk about altruism, and we can actually see there's a lot of altruism there, because as I mentioned, the workers, they are just like the colony soma, and when I say soma, that means that this worker, they have no future reproductive potential. So you see all their life is being altruist, because they are all their life is functioning for the whole colony, and so that's what I meant, that there's a huge difference between the let's that's what I meant, that why we say that the ant colony is like an animal, because you can see that in an animal you have this each cell, the somatic cell, like a muscle cell, neuron cell, they actually cannot pass on their genetic information to next generation. They can only pass on the genetic information through the germline. And so in this sense, you can see that each cell in our all the somatic cells in our body, they're actually all being altruist. Yeah. So this is that's what I think about the ant colony. Yeah.
Amazing. I love that. And I love that like last little part. This is the way I think about it. And I guess that's why I mentioned that the philosophical implications of your work is actually quite immense. And it would be amazing if you decided to come back and share some more, because hopefully that day I won't be so distracted and I'd be able to ask some better prepared questions.
Oh, thank you. Thanks for your interest. Yeah.
Oh, one question related to that would then be, what if you put different ant colonies together in one space? What happens? Do they like kill each other? Do they kind of the bigger queen and workers win, or they just work then all together? Like, has I guess that experiment has been done at some point.
Yeah, I mean, we actually when I was a PhD student in the lab, I actually did this a lot. I try to move a worker from one colony to the other colony to see what happened, and then you can see they usually being just killed. Yeah, so they are just like a really, because for example, you can see that you think about the transplant. If we try to transplant the organ from one individual to another individual, you got this immune response, because the two different individuals, they usually cannot upset the organ transplant. And similarly, when I try to transplant the individual worker from one colony to another one, this individual will actually have this will be what's that kicked out. Yeah, but of course this depends on species. In some species, in some species, this merging, let's say two different colonies, they can actually fuse together. And about the frequency, that's something that I cannot say for certain. Yeah, but I think that's something not happen very frequently. But for some species like the Monomorium and also some Argentine ant, they sometimes can fuse together. So this depends on species, I think. Yeah.
Ah, interesting. You know, it's different, but it kind of reminds me. There's one parasite of bees that is quite common here in the US, and the Asian bees recognize the parasite and kills them. Like, so the immune system is able to recognize and kill it by temperature and vibration, I think. But the bees in Europe and the US, they are not able to even recognize it. That's why they die much more, and this parasite keeps killing the colony so much. So maybe in those different species, they overlap so much that they don't recognize them as foreign. And it would be interesting to see what would be necessary to, what's their immune system, like what's their antibodies or so. What do they recognize as different? It's really interesting. Perfect. So again, thank you so much for coming. This is really so interesting. This was a real treat. And yeah, we wish you all the best for your future and your research. And hopefully one day you'll come back and update us on your current work that you're doing. So thank you. And we are looking forward to the future.
Oh, thank you. The pleasure's mine. I really appreciate the opportunity to. I'm really happy about your interest in our work. Yeah.
Yeah, thank you. And also in the comments, people are being very appreciative. So yeah, thank you, everyone. And if you like discussions like this, follow the club. We will have another room with Dr. Ando, who will show how life adapted to oxygen. And then we'll have one more room with Dr. Xi, who will talk about a new way of discovering personalized drugs based on omics data integration, which will also be really interesting. So I hope I hear you all back, and enjoy the rest of your day, morning, evening. And thank you so much, Vitam. Really appreciate it, and good luck for everything.
Yeah. Good luck. Yeah. Goodbye. Have a good evening. Or good afternoon. I don't know the time.
Yeah, everyone is in different time zones.
Oh yeah. Yeah.
Okay, I'll close the room in 3, 2, 1. Bye,
Yeah.
everyone.
Yeah.
Thank
Goodbye.
you.
Yeah.